myself Hiral Suthar. Today we will discuss about antigen and antibody. Here we will understand the chemical nature, the types, properties, structure, function and the classification of antibody and antigen. Let's first understand what is an antigen. The antigen is defined as a substance which, when introduced parenterally into the body, they stimulate the production of an antibody. The antibody or a T cell form in response to antigen and they are specifically reactive against the antigen. Any foreign substance, just like a dead or living microorganism, bacterial toxin, may act as an antigen. There are two characteristics of antigenicity. The first is immunogenicity. It is the capacity to provoke a specific immune response. The second is immunological reactivity. It is a specific reaction of antigen with the corresponding antibody or specifically sensitized T lymphocyte. Now, let's see the types of antigen. Based on the ability of antigen to carry out the two functions, they are classified into two types. The first one is a complete antigen. It means antigen which is able to induce antibody formation and is able to react with it specifically and in observable manner. It is known as a complete antigen. The second one is incomplete antigen, also known as a hapten. A substance which is unable to induce antibody synthesis by itself but can react specifically with the antibody is known as the hapten. Hapten are incomplete antigen which become a complete when they react with the carrier molecule. We can see here the hapten molecule which is bind with the carrier molecule and make a complete antigen when the immune system is induced. Hapten are a generally low molecular weight lipid and carbohydrate while the carrier molecule are a protein. Now, let's see the chemical nature of antigen. Antigen are a protein, polysaccharide, lipid or nucleic acid. Protein antigen are more effective than polysaccharide antigen. Lipid and nucleic acid are less antigenic, but the antigenicity can be enhanced by coupling them protein. Not all protein are antigenic because the presence of aromatic radicals such as a tyrosine, it is a most for antigenicity. Gelatin is an example of non-antigenic protein. Now, we will see the properties of antigen. For any substance to be antigenic, it should have the following properties. The first, it should be foreign to the host. Now, the recognition of self and non-self is an important function of the immune system. The body does not respond to a self-antigen, hence, the substances that are foreign to the circulation are antigenic and are able to mount an immune response. A substance which is more foreign to the host, it is a more powerful antigen. The second is, it should have a molecular weight more than 10,000. A substance having a molecular weight less than 10,000 are weakly antigenic or a non-antigenic. The third is, it should be a chemically complex. Substances which are chemically complex and possess aromatic radicals such as a tyrosine are a more antigenic. The fourth is, it should be susceptible to tissue enzyme. After administration of antigen into the body, it is a process by phagocytosis cell and intracellular enzyme into appropriate fragments containing epitopes. The epitopes is an antigenic determinant responsible for immune response. This initially processing of foreign substance is a must for immune response. Substances which are resistant to the processing are not antigenic. Now, we will understand what is antibody. Antibody are a peptide molecule that are secreted from a plasma cell. Two modes of immune system occur in our body. The first is a cell-mediated immunity and the another is a humoral immunity. In humoral immunity, the complement system and the antibody production occur. The difference between cell-mediated and the humoral immunity is a one-line difference. They are a macrophage, natural killer cell that protect us against the infection. If there is any infective agent, they will kill them or engulf them. But once the interaction with such of those antigen presenting the cell involved with the T helper cell, they activate B cell. Once they activate B cell, the B cell convert itself into the plasma cell. Now, the plasma cell are bigger than B cell. The small modification take place in B cell. So, it have a huge area of cytosol where they start producing different types of protein, and this protein is known as an antibody. Now, the antibody is defined as a immunoglobulins are a group of glycoprotein present in the serum and tissue fluid of all mammals. Now, the nature of antibody. Antibody are a globulin protein in nature and also known as immunoglobulins. They contain sugar residues and hence are glycoprotein. They constitute 20 to 25% of total serum protein. When separated electrophoretically, most of them migrate in the gamma region, hence they are also termed as a gamma globulin. Most of them have a molecular weight 1,50,000 to 1,18,000. Antibodies are a thermolabile and denature on heating at 70 degrees Celsius for 1 hour. Their activity is also affected by the pH of the medium and the other factor which affect the properties of protein. They are not denatured by ethyl alcohol at low temperature, but at higher temperature it can be denatured by alcohol. Natural salts such as magnesium sulfate, ammonium sulfate and the sodium sulfate can precipitate them but do not denature them. Now, we will understand the structure of antibody. Typically, the structure of antibody is in Y shape. You can see in this structure. It have a 2 heavy chain and 2 light chain. Now, with the help of specific Y shape, the structure has a region called as a FAB region. This is a hand on Y. We can see here the FAB region. With the help of FAB region, they can be specifically interact with the antigen. Okay. There is a portion of bottom known as a Fc region. With the help of this Fc region, it interact with the cell surface receptor of our immune cell. For example, B cell, lots of antibody attached to the B cell with the help of Fc region. Now, we can see in detail, there is a 2 heavy chain. This small fragment is a light chain. Now, the chain are vary same time and repeated time to time. and different class of antibody, the number of chain varies. Not the number of chain, but the repetition of amino acid in the chain is variable. This amino acid is bind with the disulfide bond, we can see here. In difference region, you can use number of disulfide linkage. For example, if you just divide it into further class, they are written as V and C. We can see here. The first fragment which are directly contact with the antigen, that portion is known as a variable region or a V region. The variable region means the amino acid sequence of this region can vary from cell to cell, time to time, and this can be a varies. That is a very important because in our body there are so many different pathogens are impacting. There are so many pathogens are involved in the pathogenic reaction. So our body needs to be prepared against all of them. We don't know what kind of antigen is going to enter. Whatever type of antigen enter, we have at least one match with the help of variable region. The rest of region is C. Here C for constant region because those part of amino acid sequence remain constant. They do not change, mutation don't take place in those region. The light chain are smaller chain having a molecular weight 20,000 to 25,000 and are attached to a heavy chain by disulfide bond. The L chain are similar in class of immunoglobulins and occur in two groups, kappa and lambda. The antibody molecule may have either kappa, the 60% of immunoglobulin molecule, or in lambda 30% of immunoglobulin molecules. Now, the heavy chain are a larger chain having a molecular weight 50,000 and are linked together by disulfide bond. H chain are structurally and identically different from each other, class of immunoglobulin, and are designated by the Greek letter as follow. The class IgG, the types of chain is gamma. The class IgM, the types of chain is mu. The class IgA, the types of chain is alpha. The types IgT, the types of chain is delta. The class IgE, the types of chain is epsilon. Now, let's see the function of antibody. They neutralize toxin and viruses, opsonize microbes to be easily phagocytes, activate a complement and prevent attachment of microbes to the mucosal surface. And catalytic function is antibody can act as an enzyme to catalyze synthesis of ozone that has a microbial activity. Now, let's understand the classification of antibody. Human sera contain 5 different types of immunoglobulin which include the following: immunoglobulin A, immunoglobulin G, immunoglobulin M, immunoglobulin D and immunoglobulin E. Now, we understand the all types in detail. The first one is immunoglobulin G, IgG. It is the major immunoglobulin in normal serum, accounting for 70 to 80% of the total immunoglobulins. It is distributed almost equally intravesicular and extravesicular compartments. It is a monomer, consists two heavy chain and two light chain. It has the ability to cross the placenta and reach a fertile circulation to provide a major line or defense against the infection in newborn for the first few weeks. It neutralizes toxin and viruses, activates the complement and enhance the phagocytosis. It participates in most of the immunological reactions. The examples are in precipitation, in complement fixation, in allergenic reaction such as Athers reaction, and also in autoimmune disease. When it is administered passively, it suppresses the homologous antibody synthesis by feedback mechanism. This property is utilized into isoimmunization of women during delivery. Now, the second type is immunoglobulin A, IgA. It is the predominant antibody molecule present in the seromucous secretion. It is also known as a secretive antibody. The secretions such as the saliva, tears, nasal fluid, sweat, cholesterol, and the secretion of lung, urinary tract, gastrointestinal tract. It is the second most abundant antibody in the normal serum. The IgA in secretion is a dimer containing 4 heavy chain and the 4 light chain joined together by J chain, which is synthesized locally by the plasma cells situated near the mucosal or granule epithelium and secretively concentrated in secretion and on mucous surface, forming an antibody paste. It plays an important role in local immunity against respiratory, intestinal and urogenital pathogens, inhibiting their adherence to this mucosal surface and thereby preventing their entry into the tissue. IgG also activates complement and promote phagocytosis and intracellular killing of microorganisms. Now, the third type is immunoglobulin M, IgM. It is the first antibody to be formed by the fetus and the first antibody to be formed by the navy B cell in response to antigen stimulation. It is a pentamer containing 10 heavy chain and 10 light chain, 5 subunits of monomer joined together by J chain, joining chain. Most of them IgM 80% in intravascular, in distribution confined to the bloodstream. Hence, it offers the protection against the bacteremia and septicemia. It activates complements and neutralizes toxin and viruses. It is the most efficient in agglutination, complement fixation and cytolytic reaction and also in immune hemolysis, opsonization and bactericidal action. Now, the fourth type is immunoglobulin D, IgD. It is structurally similar to IgG. It is a monomer containing 2 heavy chain and 2 light chain. It occurs in low concentration and is mostly intravascular. It may function as an antigen receptor on the surface of B lymphocytes for recognition of antigen and for the control of activation and proliferation of lymphocytes to produce antibodies. Now, the last type is immunoglobulin E, IgE. It occurs in a very low concentration in serum and is structurally similar to the IgG. It is a monomer containing two heavy chain and two light chain. It is extravascular in distribution and has an affinity for the surface of tissue cell, particularly mast cell and basophils. It causes a degranulation of this cell, releasing a pharmacologically active substance. This is responsible for type 1 hypersensitivity reaction. It is also play an important role in immunity against helminthic parasites. Thank you.